Hi guys, welcome to anatomy lesson 7.04 on the anatomy of myofilaments. Just when you thought we couldn't memorize anything else about a muscle cell, turns out we still need to talk about the anatomy of these myofilaments because that's how down into the nitty gritty we're going to get. So let's just look at our overview here. I've got a muscle fiber, which is the same thing as a muscle cell. These are pointing to nuclei. I've got in yellow the T tubules and in green the sarcoplasmic reticulum. It's not labeled, but off to the sides. I would have the terminal cisternae. Hopefully you have that memorized by now. So if you don't have some of these terms down, it's going to get very sticky. Then I have a single sarcomere, and notice it goes from Z line. They say disc here. I've never seen that before, but we've got Z line to Z line is a single sarcomere, just like we said in the last video. And then I have a dark area for my A band. So this is going to be my A band. And I have I bands that are lighter. In the middle of the I bands, I have my Z lines. Let's just cross that out and put a line. And the A band has two different areas of darkness. This is so we have lighter, next lighter, and then so darker, lighter, and the lightest. So here we have an overlap. The darkest part of the A band, we have an overlap of actin and myosin. In the middle, we have just myosin with a slight line through the middle called the M line, and this is going to be called the H zone. All right, so we said in the last video, this isn't really anything new, where we have this myosin, and these are those cross bridge heads coming off. So the only thing you really need to know about myosin is that we have cross bridge heads. And it turns out that there is a site on actin, and I'm going to highlight it in red here. It lies just underneath these three green beads. So actin is really what we want to talk about. And that little red line that I put on there is the binding site for the myosin cross bridge heads. But that stays covered up because if that was open, what would happen is we would just perpetually have a binding and things would never be able to relax. And so we have to keep that covered until we need it to be available. All right, I'm going to try to redraw what we have over here. Actin is actually two protein strands. So we've got these really thin strands of protein that kind of loop around each other like this. They kind of think about if you had two strands of pearls, and they do occur as these little, it just takes too long to draw out all the little circles. But if I was going to draw out actual actin, I would want to draw it out like this. I have a third strand. So we've got two strands of actin. So this is actin. And this is actin. But then our third strand is another protein called tropomyosin. And I'm going to do this in dark blue. So it kind of comes in and wraps around the other two in dark blue. And attached, so let's say we've got a binding site right here. So there's a binding site. This tropomyosin has on it a complex. We're going to do these of 3 round proteins. 3 round proteins. And these round proteins are called the troponin complex or just the troponin. And when the cell is at rest, so at rest, the myosin is not bound and the binding site is covered by troponin. We also need to know that that troponin is held in place by the tropomyosin. So when we get ready to create a bond between the actin and the myosin, this troponin has to move, and it's the tropomyosin that does it. So the tropomyosin actually slides. We're going to see it slides out of the way, and it opens up that binding site. Well, what's going to cause it to move or go back in place? It's going to be calcium. So we've talked in the past about the fact that calcium is really, really important for muscle contraction and other things. And part of that reason is because calcium controls opening and closing those binding sites. All right, so our takeaways for this video, we're finally done with the anatomy. We don't have any more anatomy to learn. We went back through the anatomy of a sarcomere where we have the A bands and the I bands, I bands and A bands. The whole thing is a sarcomere from Z line to Z line. We've got a lighter zone in the middle, which is the H zone, and the M line. And the colors are formed by the overlap. But then we got into the anatomy of the myofilaments. So myosin, thicker, it's got these cross bridge heads. This really thin actin has 2 strands of actin and 1 strand of tropomyosin twisted together. And that everywhere there's an active site, tropomyosin has these 3 round proteins called troponin that cover up the binding site. And so we're finally ready to start to talk about muscle contraction. We're going to start off with a big idea, and then we're going to work our way down to the details. All right, that's all for now. Thanks for watching.